friends, welcome to Datta Biology class. Let us continue our discussion on transcription. In the earlier episode, we have discussed about amino acids, how they are and how they are joined one with another. Now our discussion will continue on RNA. Actually, from DNA to RNA, it is known as transcription. And from RNA to protein, synthesis is known as translation. And in molecular biology, this is known as transcription. This is known as translation. So from DNA to RNA it is known as transcription, and from RNA to protein synthesis is known as translation. And the entire thing is known as central dogma. Central dogma in molecular biology. Why we are discussing this? Because we are producing protein. So it is a case of protein synthesis, I told you earlier. Because of this synthesis of protein, these steps are being taken. Now, from DNA to RNA, RNA is produced, and in this RNA, we are producing mRNA, we are producing tRNA, and we are producing rRNA. So RNA of all types are being produced from DNA. And our discussion for transcription will be confined to mRNA. This is known as transcription. And these are only RNA synthesis, tRNA synthesis, rRNA synthesis. But producing mRNA from DNA is known as transcription. Okay? Up to this clear? Now, RNA of all types, that is tRNA, mRNA, and ribosomal RNA are produced from DNA. Let us suppose this is DNA, this is uncoiled. From this DNA, we have replicated, in case of replication, from this DNA double strand, two double stranded DNA has been produced, that is replication. And here we are studying from this DNA strand, RNA will be produced. So, suppose this is 3' to 5', and this is 5' to 3' direction, the two strands are there. They are in coiled condition. I am showing here as an uncoiled condition. Now here, nucleotides are arranged. Here nucleotides are arranged. In this way. So from these nucleotides, RNA will be produced. How? The entire DNA will not produce RNA. Because the entire DNA, if replicates, it becomes replication. Now we cut a simple, say from, for example, let us suppose from this portion to this portion, RNA will be produced. So I will produce RNA from here to here. So this RNA will be produced in short pieces. And the same RNA, now this is for say mRNA. From this portion, DNA can produce tRNA. From this portion to this portion, DNA can produce ribosomal RNA. So it is the DNA which directs the production of mRNA. Now, we know that it is a cell. Cell has nucleus. So all the RNA, that is mRNA, tRNA and ribosomal RNA, are produced within the nucleus. And then after production of this, they come out from the nucleus and they go to their respective places. Now mRNA will join with ribosome, the protein factory. And this tRNA will be there in the cytoplasm. And rRNA will be attached to ribosomal RNA. Okay? So, all the RNA are produced from the same DNA strand. So, DNA strand, it may be called that this DNA strand is the template strand. We can say, because of this DNA is giving direction that some type of protein will be produced. They will produce protein as per the direction of DNA template strand. So from the DNA template strand, RNA will be produced. We are confined to mRNA now, not tRNA, rRNA. So here, one strand will be produced in this way. This strand, this will go in 5'3" direction. So this strand will be called sense strand, and this strand will be antisense strand. This strand will be called template strand. This is non-template strand. Okay. And this region from where this mRNA is produced, is region is known as coding region. These are the tags. Now, here we will produce mRNA. So these three will nitrogenous bases will be like this. Clear? So these nitrogenous bases will be complementary to these nucleotides or these bases. So if it has A, T, G, C, A, T, G, C, A, T, G, C, A, like this. If these nucleotides are here in this way, so we will produce, the mRNA will be produced, will be complementary to that. So A will be replaced by AT, T will be A, G will be C, C will be G, A will be T, T will be A, GC, A, T. The irony is that here, if it is produced in this way, it can be called DNA, because it is single strand and moreover it is RNA. So what we have said that thymine will be replaced by uracil. Thymine will be replaced by uracil. Thymine will be replaced by uracil. Clear? So this is RNA, and it is complementary to the DNA template strand. And this is the mRNA. It will start out in short pieces. In prokaryote and in eukaryote also, these are the DNA and RNA are synthesized with the help of RNA polymerase enzyme. Now, in eukaryotes, there is nuclear membrane and there is nucleolus. These are advanced eukaryote. But in prokaryotes, this is a cell, and within the cell and DNA are there. So, in prokaryotes, this transcription occurs in cytoplasm, and in eukaryotes, this transcription occurs in the nucleus. Both are aided by an enzyme known as polymerase. Those are known as polymerase enzyme. And since RNA are producing, so they are called RNA polymerase enzyme. Okay. Now, this is being produced under the direction of who? Who is the director? Director is DNA. Because DNA in the nucleus, DNA is itself instructing its own strand to produce this mRNA to produce some protein. Because out of this, some protein will be produced which will come later on. So it is the DNA directed RNA polymerase. So people are called DNA dependent RNA polymerase. So this is known as DNA dependent RNA polymerase, and in short it is known as RNA polymerase. Now, in bacteria and or in prokaryotes, see the difference. In the prokaryotes, there is no nuclear membrane, there is no nucleolus, and other it is modern nucleus. It is old nucleus, it has only DNA strands. So, in E. coli and other in bacteria, the DNA is circular in nature. DNA is circular. So this is the DNA in prokaryotes. In prokaryotes, difference you can easily understand. In prokaryotes, suppose this portion will be converted to from this area to say this area. From this area to this area, RNA will be produced. So it is within the cytoplasm it is being produced. This RNA will be complementary to the this strand, will be complementary to the this strand, and this strand have nucleotides arranged in this way. So in prokaryotes, the single RNA polymerase can produce this. This single RNA can also produce, say from here to here, they can produce another RNA. They can produce another RNA. From here to here, they can produce another RNA. And all those are complementary. Those are complementary to the DNA nucleotides. To the DNA nucleotides. So, in a single RNA polymerase, can produce mRNA, tRNA, and rRNA. So, in prokaryotes, the single RNA polymerase can produce all these three types, all the RNAs. As simple as that. Now, in eukaryotes, there are three RNA polymerase. RNA polymerase 1, I am writing RNA polymerase 1, RP1. I am writing RNA polymerase 2. I am writing RNA polymerase 3. It has been seen that in this is known as eukaryotes. In any eukaryotes, there are three RNA polymerase. RNA polymerase 1, 2, 3. Now, difference is that RNA polymerase is produced within the nucleolus. This is known as nucleolus. RNA polymerase 1 is produced in the nucleolus. Clear? And RNA polymerase 2 and RNA polymerase 3 produces ribosomal RNA and transfer RNA. So, transfer RNA is produced within the nucleus but not within the nucleolus. That produces within the nucleus, not in the nucleolus. So it is in the nucleolus and this is in the nucleus. 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 These are produced in the nucleus and this is produced in nucleolus. This produces tRNA. This produces rRNA. This produces rRNA. This produces messenger RNA, polymerase 2, and this is messenger RNA and transfer RNA, tRNA. Write it down. Write it down. Please write it down because sometimes we may forget. So it is in the eukaryotic cell, the RNA polymerase are three types. RNA polymerase 1 produces rRNA and it produces within the nucleolus, and these two are not produced within the nucleolus but within the nucleus, within the nucleus, not in the cytoplasm. So these two are produced, that is mRNA and tRNA. So I am writing here mRNA and tRNA, and here I am writing this is ribosomal RNA. You write it down here rRNA. Now it is clear. So these three are produced within the nucleus. And during the production of this mRNA, tRNA, rRNA or whatever RNA, during the production of this, RNA polymerase helps. Now there are several factors. There are factors like initiation factor. There are factors like initiation factor. Let me write it down here. Initiation factor is denoted or represented as IF. Initiation factor 1, initiation factor 2, and initiation factor 3. But these factors are themselves are protein also. The irony is that these factors are themselves in proteins, and they help for transcription process initiation factor. Transcription takes place in 3 steps. We know initiation, elongation, and termination. These are the 3 steps, very easy steps, which we used to do, but we do not go into the details of these. So in initiation factor 1, 2, 3 are there, but in prokaryotes as well as in eukaryotes, initiation factor 2 takes place. 2 helps. So initiation factor 2 helps in initiation in both prokaryotes and eukaryotes. And then elongation factor is known as elongation factor 1, elongation factor 2. So elongation factor helps for elongation of the polypeptide chain. This much we should know. We can go into the detail, but that is not that much required. There are some other factors like initiation factors and elongation factors helps in the process of transcription, which are called elongation factor 1, 2, 3 and elongation factor 1 and 2. There are also TU, TS, so many things are there. We will simply confine to elongation factor and 2. Elongation factor 2 for elongation, usually elongation factor 2 is required. Anyway, now for this transcription, we have come across, first we have come across amino acids that we have gone through. Now we are coming to mRNA. First we will discuss our discussion on mRNA. mRNA, we have discussed that mRNA, in mRNA, the process of transcription starts with initiation and this. There are some factors which are coming. Some sigma factor, rho factor here. And here no factor is required, will come slowly. But because of the transcription, some protein factors, they themselves are protein, they helps in the process of this, and initiation factor 1, 2, 3 helps in initiation of this chain in this process, and for elongation factor 1 and 2 are required. Okay. Now in prokaryotes, we have seen the RNA polymerase is capable of producing mRNA, tRNA, rRNA with the help of single RNA polymerase. But in case of eukaryotes, RNA polymerase 1, RNA polymerase 2, RNA polymerase 3 helps for the production of, this is known as mRNA. mRNA is polymerase 2, this is polymerase 2, this is polymerase 1, and this is polymerase 3. We will go into the detail of tRNA. And for ribosomal RNA, we will go into the detail of ribosome when we will come to ribosome, that what actually ribosome does. Now, from whenever it is produced within the nucleus, this goes out to cytoplasm or cytosol. So all these RNA, mRNA, tRNA, rRNA, they come out of the nucleus or from the nucleus to the cytosol. mRNA attaches itself to ribosome. Here ribosome will go. Ribosome are made up of 2 parts. This is a one part, another part is like this. Bigger and smaller subunit are there. This subunit will go through. So mRNA comes here, Ribosomal RNA comes here and tRNA remains in the cytoplasm. This is tRNA remains in the cytoplasm. Here tRNA remains in the cytoplasm. mRNA attaches itself here for protein synthesis. mRNA comes here, sits here, attaches here for the purpose of protein synthesis, which will come, and ribosomal RNA is here. Within the ribosome, the other ribosomal RNA. So it is the DNA which first sends or instruct the ribosome to get prepared by producing ribosomal RNA. Then DNA gives instruction, DNA dependent polymerase gives instruction to produce tRNA. So it is giving instruction to tRNA for its specific function to collect the amino acids available in the cytoplasm. That will be done by tRNA. And last, mRNA will be produced when you go there, sit in the ribosome and produces the protein. So in every moment there is direction from DNA. So it is DNA dependent. Now we will go, the nucleotides of mRNA are complementary to the DNA nucleotides. Only exception is that T is replaced by Q and the sugar is replaced by, sugar is, this is deoxyribose, this is ribose sugar. We have carbohydrates. Now the strand, how does it, this strand, how does it make the process? What is the actual process? And prior to this process, we will go, we'll go there. We'll go there. What actually happens? DNA strand, it remains uncoiled. Actually, during the production of M, we are confined ourselves now on mRNA, not others. This production of mRNA, the DNA identifies that mRNA is what for it is being produced, because mRNA will come here and produce protein. Protein, I have said that protein is, say for example, protein is Maggi. Protein is Maggi. This production of protein will be done here. And this production, it is giving instruction that I want some hormone to be produced for X and Y Z purpose. I want this enzyme, pepsin or pepsinogen. So I want this tyramine, amylase, whatever it is. This protein I require. So it, because of nucleus is giving instruction, who is giving instruction I don't know, but DNA is having that instruction that we have to produce pepsinogen. Say for example, pepsinogen. So DNA identifies the places where from it will start. So DNA, there should be an initiation point, say for initiation point here. Initiation point. In prokaryotes, the matter is rho factor and sigma factor. Okay, let us complete that first. So in prokaryotes, this DNA so this DNA instruct here that from here to here I am giving an instruction that mRNA will be produced. So this is the initiation point, and this is the last point. Who will decide that this is the initiation? This is this. This is done by here. I'm writing it here. There is a region called promoter. Promoter is not anything. Promoter is the region of this. This is the promoter region. Here the initiation point, that is from here, the cut will be there, production will be there. This to this production will be there. Same. So here mRNA will be produced from here to here. So this is the start point, say this is the end point. Who decide? This end point is decided by rho factor. Last. And this point is decided by sigma factor. How does it happen? This sigma factor RNA polymerase along with sigma factor plus sigma factor. This is known as sigma factor. RNA and sigma factor sits here in the promoter region. RNA and sigma factor, this is the initiation point from where it will start. So these two are called hollow enzyme. And without this is known as core enzyme. So this hollow enzyme comes here. It already got the initiation point, starting point here. From this starting point, it will start the complementary strands. Complementary strand to that of the DNA. DNA has complementary, DNA has this up to this, and it will produce the complementary to that. Only the thymine will be replaced by uracil and from deoxyribose to ribose. So it is a sigma factor in prokaryotes, not in eukaryotes. In prokaryotes, sigma factor and RNA polymerase produces hollow enzyme, and this sits in the promoting region, and they find out the starting point. Sorry, not here. They find out the starting point from where it will start. Usually it has been seen that here AUG, while we are finding AUG, deoxyadenylic acid, uracilic acid, guanilic acid, wherever they will find AUG, it will produce methionine, AUG. Here it will be because of the A replaced by T, U is equal to G, G is equal to AU, G is equal to C, U is equal to A, sorry, TSC. So here the point, the starting point will be here. Usually methionine, in all the protein synthesis, you can see that methionine is the first production of amino acids, and from methionine RNAs are produced in a definite sequence for production of protein in this prokaryote. But in eukaryotes, we are coming now. In eukaryotes, so this is DNA. DNA is double-stranded DNA. This DNA directs itself, direct a portion of itself to produce protein. So for the purpose of production of protein in ribosome, this DNA is giving instruction that this protein or pepsinogen will be produced. So it decides the place, the initiation factor. Usually initiation factor starts with AUG. So it starts from, say for example here, this is 3' to 5', because mRNA starts from 5' to 3'. mRNA starts from 5' to 3'. From here it starts like this and it ends, say here. This is 3' to 5'. Okay. Starts here, ends here. In eukaryotes, here is a point called initiation point, decided by DNA itself. That this is the initiation point. From here you start, here you end. Usually starting point has AUG and it has got 3 stop codon. Stop 1, stop 2, stop. Will come. What are the stop codon? What are the methionine is the initiation codon? Will come later. Then it will be easier for you. Now this mRNA is produced. How? First you have to uncoil. So the DNA in the DNA, the coiling gets loosened and it opens. So coiling opens here. Coiling opens like this and this coiling here like this. Like lubrication bubbles. So they uncoiled it and mRNA produced. It is complementary to the DNA. The DNA giving instruction to its nucleotides that you start here, end here, and it accordingly it starts here. And it is ending here. As I'm going this uncoil, it is being produced, mRNA is produced and followed by recoiling of the DNA. Once it is produced, it again recoiled as its original form, and this mRNA is produced within the nucleus, and from the nucleus it goes out. Here, now this portion is known as, this portion, from this portion to this portion is known as coding region. Coding region. This portion is known as, sorry, it's not visible. Coding region, this portion is coding region. REGION, coding region. And it is 3' to 5' and it starts from 5' to 3'. It is called template strand. It is called sense strand. And the other one, this is sense, this is nonsense or antisense. Antisense strand. Here, these are the terminology. If you go through the books, you will find it difficult. Now, what will be template? What will be this? Simply, the strand, you write, it is coding strand. And this is the antisense strand. Or this is sense strand, this is antisense strand. Only single nomenclature you keep it in mind so that you may not puzzle during examination hall. This is sense strand, this is antisense strand. But this will solve the purpose. Now we will go to tRNA. mRNA we have produced. Now we will go to tRNA. It is known as transfer RNA. mRNA brings the message from DNA that this protein will be produced, which will be complementary to the template strand, and this will ultimately will be translated by mRNA. Okay. Now, DNA has given the instruction to tRNA. There are huge amount of tRNA. tRNA are the smallest RNA. tRNA, RNA is single strand. So this is RNA single strand. The single stranded RNA, it is the smallest RNA, and you know this has the nucleotide. These have the nucleotides present here, because this is the RNA. It has nucleotide sequence of 74 nucleotides to 90 nucleotides. So tRNA is present in the cytoplasm on being produced from the nucleus of the cell on the instruction of DNA, and from the DNA strand it goes to cytoplasm, and it is the smallest cytoplasm. It remains within the cytoplasm. It is like a single strand, and it has nucleotides 74, 90 nucleotides, but we are still calling it as a smallest RNA. Clear? Now this RNA, these are the nitrogenous bases. They have the affinity to join with the other, so it remains joined in some places where this is the structure of tRNA. Two-dimensionally it is seen in, you have come across, it is like clover leaf. In clover it is like this, clover leaf it is like this. So it has three bubbles. Okay. Now since it has three bubbles, three places are there, and the nucleotides lies here. Nucleotides lies here. Is that clear? Nucleotide lies here. So here is one nucleotide, here is one nucleotide. Here is one nucleotide. Here is nucleotide. So it may 1, 2, 3, 4. 4 region joined by hydrogen bond. In DNA we have come across that it remains joined by hydrogen bond. Hydrogen bond A, T and GC. GC 3 hydrogen bond, ATGC. So it remains joined in 4 places. 1, 2, here hydrogen bonds are there. It remains joined because it cannot stand in this way. It cannot stand. So it produces 1, 2, 3, 4 hydrogen bonds, bonded region, and it is in the form of clover leaf. Now this portion is 3 prime, this portion is 5 prime. We are discussing on tRNA. tRNA is available in cytoplasm. Now this is in the form of, tRNA is in the form of This clover leaf, it has 7492 nucleotide residues, and in 4 places it is seen that it makes some bond, hydrogen bond with nuclear bases, nucleotide bases remains in this way. And its 3' remains here, the CA. These bases are there, CCA, cytosine, adenine. And 3' is hydroxyl ion at its tip. How it is 3' hydroxyl bond, hydroxyl at its tip? Usually the question may come. So let us suppose C1, C2, C3, C4, C5. This is sugar, here also sugar, and 3' is equal to C1, C2, C3. C3 has OH has its tips, and 3' is here, 5' is here, 3, 4, 5' is here. Okay? This much I will go, not further. So this is the 3' and this is at the tip, it got OH at its tip. Now what it does, this tRNA, there are 20 amino acids. These 20 amino acids are here in the cytoplasm. Clear? These 20 amino acids will be collected by this transfer RNA. It will transfer, it acts like the packers and movers. It pack the amino acid, will come during the transcription process. Amino acid will join with amino acid. It join with amino acid. So there will be 20 type of amino acids. There will be 20 minimum tRNA, and it will go and produce protein. Will come later. So this tRNA has this portion. This portion will collect the amino acid. Say amino acids. Name any of them. Leucine, isoleucine, lysine. Say leucine. Leucine, the amino acid in the cytoplasm. Leucine will be here. So this site is known as number 1 amino acid binding site. This is known as, this portion 3' is known as, in tRNA this 4 portion has been identified. This is amino acid binding site, number 1. Number 2, here tRNA have codon here, and it will collect the mRNA. mRNA have codon here. mRNA have codon, say A, and tRNA will have T. Correct? Not correct, because this is RNA. Thymine will be replaced by uracil. So here it will be U. This is called codon because this is in mRNA. This is called codon. Codon triplet. This 3 actually produces triplet. We will decide, because RNA to protein will take another episode. So this is known as codon, and this portion is known as anticodon. anticodon. So we have got amino acid binding site and this is anticodon site or codon binding site. Codon binding site or write anticodon site. It will be convenient. Anticodon site, number 2. Number 3 and number 4. Here is remain attached with ribosome. So number 3 is ribosome binding site. Ribosome binding site. It attaches this tRNA. This tRNA attaches with the ribosome here. And this is question. This is another question. tRNA joins here. So it is known as ribosome binding site. And here, because tRNA attaches the amino acid with the help of amino acid synthetase will come. So what enzyme helps here to bind it? That enzyme question remains here. This is known as enzyme binding site. So this is known as enzyme binding site. So in tRNA we have come across everything that tRNA having this site and it has got CCA in the 3' end with the hydroxyl at its tip and it is the amino acid binding site. This is amino acid AA. Amino acid binding site. This is anticodon site. It will produce anticodon like this. Suppose AAA will produce UUU. tRNA has the capacity that UU will be produced. Then leucine or glycine or that will come. We'll see that what amino acids will be there in case of UUU. In case of UUU, let me say, let me go through. UUU is phenylalanine. UUU is phenylalanine. Phenylalanine. So this is one amino acid. So there should be minimum 20 amino acids. There should be minimum 20 tRNA. There should be 29 amino acid synthetase enzymes, which will produce the protein, which will help in producing protein. Because in order to produce protein, not mRNA is alone. mRNA with the help of ribosomal RNA, mRNA with the help of transfer RNA, they produce, these three produce protein. That's why DNA first produces, gives instruction to tRNA, then it gives instruction to ribosomal RNA, then it produces messenger RNA. So in this order, DNA produces these three and they ultimately produce protein. This is known as tRNA. So we have come across tRNA, mRNA and ribosomal RNA will come in the next episode. Till then, take rest. Thank you very much.